GYN cancer field, I think it's an interesting ASCO. There isn't any one dominant trial or theme, and many of the areas of interest are beginning to mature, meaning that we're getting more data to put them in perspective. So what I mean by that is combinations of PARP inhibitors and immune oncology drugs are now being presented, giving us a better idea whether that combination is going to work. And we're seeing the maturation of some fairly large phase 3 trials like GOG 213 to tell us a little bit about whether patients would benefit from surgery. So it's a broad spectrum of studies that are fine-tuning the way we approach our patients. The clear changing landscape is it involves PARP inhibitors. So obviously we know that SOLO2 and NOVA trials are out, leading to approval of those agents, and now Ariel 3 has been described and gotten in front of the FDA, and the FDA has now approved rucaparib also for maintenance therapy in first-line recurrent ovarian cancer. I think the trend on that will continue in that towards the end of the year we should hear about SOLO1, which is maintenance at frontline, and PRIMA will follow right behind that. Most of us suspect or expect that those trials will be positive. And so PARP inhibitors are going to come all the way up front in terms of maintenance. I don't really believe that they'll be used in combination with chemotherapy because it's a difficult combination to get around toxicity. So that's changing. And then the other not trivial issue is that Genentech Roche is in front of the FDA with bevacizumab for approval in frontline therapy. Again, most of us expect them to get approval. So Bev will now move up front concurrently with chemo and then in a maintenance phase. All of those are in some ways game changers because once Bev moves to the front line, then you'll probably see less usage of it later on. And it'll raise issues about can we get insurance coverage for Bev after Bev. Now, there is an interesting trial presented here we call it the Mito-Mango study. It was done in Italy, which shows that if patients are retreated with bevacizumab, they benefit. It's not an FDA registration trial, so whether that'll be sufficient to provide evidence for coverage for Bev after Bev, I don't know. We'll find out. And then, as I said, PARPs will be moving up front too. So this sets the stage for really critical decisions in the field. When you see a patient, do they go on PARP maintenance or do they go on bevacizumab maintenance? And then ultimately, combinations are going to be important.
So Bev is here to stay, I would assume?
Yes, here with us. You probably know that it'll come off patent, I believe, in about 2 years. And there are several biosimilars already lining up. So the good news there is that we might see some price reduction, and there'll be a number of choices in terms of the actual molecule we use, and that's all good for patients. So I'm of the belief, looking really carefully at the randomized phase 3 trials, that from an efficacy standpoint, the 3 approved PARPs are roughly the same. The hazard ratios for instance, germline mutated patients is all around 0.18 to 0.2. For HRD patients, somewhere around 0.3 to 0.4. And then for the so-called wild-type patients, meaning normal BRCA1 and BRCA2 genes, HRD negative by assay, the hazard ratios are 0.58, whether you look at niraparib or you look at olaparib or rucaparib in Ariel 3. So I think from an efficacy standpoint, they're very, very similar. The differentiation, if you want to call it that, comes from the toxicity profile. And there are some subtle but notable differences. There are class effects, which includes nausea and fatigue. That extends over all PARP inhibitors. But for niraparib, there seems to be a more profound effect on the platelets. In fact, the indication, FDA indication, requires weekly platelet counts for the first month because of that. And then rucaparib does have an unusual effect on transaminases and even creatinine. This is not a toxicity of the end organ. There's no liver toxicity. There's no kidney toxicity. It's actually related to the pumping of the enzymes. And so these can accumulate. So people need to know about that so they don't go doing these massive workups or dose reduce when it's not necessary. Yeah, no, I think they will, undoubtedly. So obviously you have an indication in breast. I think the other PARPs will. When you have indication for one PARP, I think you'll see the other PARPs enter into that area. There's certainly homologous recombination deficiency in a subset of breast cancers. But the really interesting data, I think, is in prostate cancer. Historically, when sequencing was done on prostate cancers, there was very few germline, or there was very few somatic mutations noted in DNA repair enzymes. But that was radical prostatectomy specimens. That was the primary tumor. It turns out now, if you look at the metastatic recurrent tumor, it looks like HRD is present up to 30% of prostate cancers. So I think there's a real possibility you could see approval for these agents in prostate cancer. I think you also see, you may see some approval in pancreatic cancer. We know that patients, men who have a BRCA2 mutation are at increased risk for pancreatic cancer, and so there's certainly a subset of tumors that have a homologous recombination deficiency and may benefit from PARP inhibition. And certainly that's a tumor where the bar is very low for active agents. So yeah, I think more to follow on that in terms of more extensive sequencing. You may even see basket trials where we simply ask the question, does your tumor have HRD? And then put those patients on trial and see if there's any activity for a PARP inhibitor. There's really two major directions that field is going in. One is combining PARP with anti-angiogenic agents. I think there's a modest scientific rationale for that. Under the conditions of hypoxia, a pseudo HRD is created where cells have trouble repairing their DNA. So one could theorize the combination of an anti-angiogenic, which makes the tumor hypoxic, would then synergize with a PARP inhibitor. And so there are multiple combinations right now in trial combining BEV plus PARPs, and multiple trials ongoing combining cediranib, which is a TKI, thought to be an anti-angiogenic agent, with PARP inhibition. We don't really have any early data to hang our hats on right now. But I think those trials are going to be really interesting to follow. I favor the BEV combinations because cediranib as a drug has been pretty tough to tolerate. We conducted the phase 2 trial when I was up at Harvard, and it was a drug that caused enormous amounts of fatigue and diarrhea. The other combination getting a lot of attention, you could predict, which is IO plus PARP. And there the theory is that because ovarian cancer cells have a defect in DNA repair, they must have a lot of mutations. And those mutations create new antigens. And so this should be an immunogenic tumor. And so combinations of anti-PD-1, anti-PD-L1 with PARP inhibitors are ongoing. In fact, several companies essentially leapfrogged and brought IO all the way up front in ovarian cancer in large randomized phase 3 trials, which I thought was pretty gutsy because there's not a lot of data right now that IO works at all. The single agent response rates are around 9%. Keynote 100 just was presented at this meeting and the response rate was 9%. So we don't know where this is all going to settle out, but there is some rationale combining IO with PARP inhibitors, and you'll see more of that play out in the next year or two. And it's a really good question, and not one that I can rationally answer because we don't really know. You know, if you look at the single agent activity, as I said, it was 9% response. But one thing that's interesting about it is the duration response is pretty long. So it suggests that there's a subset of ovarian cancer patients who really do benefit from IO. but we have no idea how to identify them. You look at you can look at PD-L1 expression, you could look at mutational load. They don't seem to track really well in terms of a biomarker. So in that context, the question you ask, when would you use a combination in the natural history? I don't have an answer for that. I guess the only thing that would track for me would be not the IO part; it would be the PARP part. And as I said before, I think PARPs are moving earlier and earlier in the treatment of ovarian cancer. So if you had an IO and a PARP that looked good, it might be in first-line maintenance. So there's no issue that PARP resistance exists. In fact, you could argue that essentially everybody will become PARP resistant at some point. There may be exceptions to that because there are about 11 patients who are still on study 19. They're out about, I think, 10 years now. And if you talk to Jonathan Letterman, he kind of mumbles, well, maybe they're cured. I'm not sure. The vast majority of patients who benefit from PARP will eventually relapse with PARP-resistant disease. And so 2 to 3 years from now, we are going to have unfortunately, armies of ladies who are PARP-resistant. We need to know how to handle that. And the first step in trying to handle that is to understand the mechanism. We don't know that yet. There are there's preliminary data. There are multiple mechanisms to PARP resistance, PARP inhibitor resistance. One is you can get reversion mutations, which basically revert the mutation in the BRCA1 or BRCA2 gene. So you get a normal gene. And of course, you have a normal gene, PARP inhibition won't work very well. You can get second mutations in the same gene, which sort of partially refolds the protein. So you get partial or hypomorphic activity. That also can create resistance. There's some evidence that P-glycoprotein is a mechanism of resistance that pumps out the PARP inhibitor. And then finally, the process of DNA repair is fairly complicated. And so it involves multiple proteins. So if you're missing BRCA1, BRCA2, you can't repair your double-stranded breaks. That process is dependent upon other genes. And if those genes are missing, you then become resistant to PARP inhibition. One mechanism of resistance along that line that I am quite interested in is that there appears to be some ATR-mediated resistance. And as you probably know, ATR inhibitors are coming into the clinic. So would it be rational to combine an ATR inhibitor with a PARP inhibitor? And the answer is yes. And actually, we just got funded from a grant from the Department of Defense to do that. Yeah. So the refractory patients are the ones defined as having progressive disease or persistent disease after platinum therapy. So it could even be up front. Patients get the bulk, they get chemo, and by 6 cycles, they either have growing disease or they have clearly obvious disease. Those are refractory patients. They do extremely, extremely poorly. In fact, they do so poorly, we usually don't have enough time to put them on a clinical trial. So we don't know what to do with them. And so it's a very, very much an unmet need. There was data presented here with the Tapatio study, which I think overall isn't that interesting. It's a response rate of about 22%. We don't get too excited about that. But if you tease out the refractory patients, those patients who got that combo is niraparib plus pembro, there was a significant response rate, and you'd expect the response rate to be 0. So maybe that combo is going to be helpful for this really sick group of patients for which there is nothing. The platinum-resistant patients, we call that acquired resistant. That is an unmet need, obviously, because all patients will eventually develop it and die from platinum-resistant disease. But at least we have some choices. I mean, single-agent Doxil, Taxol plus Bev. These are regimens that we use all the time. I think you'll see new agents coming into that set of patients. The one that comes to my mind, partly because I'm working on it, is mirvetuximab soravantine. Is clearly an agent that is very active in platinum-resistant patients and is in a randomized phase 3 trial which just closed. We'll know that story in about a year. I mean, I'm not so sure the answer to that has changed radically in the last 10 to 15 years because it really relies on just a good a good clinical trial design. A good clinical trial design means that what you're offering the patient is as good as standard of care, but potentially better. If it isn't, the trial is not a correct one. And then on top of that, I think picking regimens. Obviously, there's got to be science behind the regimen, science behind the combination, preferably good preclinical experiments with results. But in addition to that, I think clinicians need to be very cognizant of convenience. And this has come up recently as we get more choices and more of these combinations. Is it really reasonable to think of a triplet or a quadruplet as a maintenance therapy? That strikes me as a bridge too far. Lots of visits, lots of infusions. Is that really what a patient wants for a maintenance therapy? So I think all of those ideas, good scientific rationale, and a convenient regimen is really important. Yeah, and it's again a really great question. We're talking bevacizumab, we're talking PARP inhibitors. Most of these are running between $12,000 and $15,000 per treatment. Not something you're going to export rapidly into a lot of developing countries. So we need to be worried. We need to be cognizant of that also. I think though that significant impact can be made and should be made because within the GYN field, some of these tumors and these cancers are differentially where there's a huge disparate impact of these in those kinds of countries. The best example is cervical cancer. This country, it's hard to get companies even interested in conducting randomized phase 3 trials in cervical cancer because we have about 7,000 deaths per year with about 12,000 cases. That's a pretty small market, to put it bluntly. But with the right trial design and rationale from scientists, lobbying industry partners, and then going to countries like India, China, where it's a huge problem, we can make a huge impact on those populations if we bring, for instance, IO to a disease which is clearly virally induced, right? So I think you'll see a lot of activity in that. Probably those trials will need to either be subsidized by the industrial partner or by governments to get them off the ground. The flip side, though, is that while it might take for a really long time to accrue to a cervical cancer trial in the United States, those international trials accrue incredibly fast. So I think we make a lot of impact on that. There are, repurposing drugs, ones that we have sort of forgotten about that we could use in a third world that might be more effective. So there's a lot of things that we could do. Well, I think the best example of that challenge is germline testing. So, now the recommendation for ovarian cancer is that every ovarian cancer patient should be sequenced. Doesn't matter the age or the histology, everybody should. And that should be done both tumor and germline. In this country, despite ASCO, SGO, NCCN, IGCS all recommending that, we still only sequence barely 50% of our patients. So we even have trouble in this country. But when you get out into Africa, the Far East, those numbers drop off radically. And part of the problem is cost, but part of the problem is just follow-up. So patients come in, they get their diagnosis of ovarian cancer. They might go back to the local community and never be seen by the academic center where they had the surgery. So that sequencing is not done. So I think for sort of biomarker and testing, which is obviously important for our newer therapies, that's a real challenge because it relies on much more infrastructure to get high quality biomarker analysis done, and that's going to take time to do that. Well, there's two points I would make on that. So first of all, the global point is no, I'm I don't think any of us are completely satisfied with both the quality and the quantity of information transferred from academic centers to the community. And part of that, it goes both ways. Part of it is because those in the community are busy and they don't have a lot of time to absorb all of the details of the new therapies. And then those of us in the academic world, we live in our ivory towers a little bit, and we read our journals, and we sort of feel that if we understand it, everybody should. So that's two weaknesses of it. The solution, of course, is courses, CME credits are really important for spreading the word on new developments. The more I love to participate in those, and part of it is because I consider it a service. We're trying to get that information coming out of the clinical trials, coming out of Journal of Clinical Oncology, the New England Journal, get it out there in an interpretable way for both patients and physicians. Second point I would make is GYN is a little special, I think, because the vast majority of the field is still managed by gynecologic oncologists who are basically surgeons. They are boarded to push chemo, but it's a very unusual circumstance. There are no other surgeons who can push chemotherapy. There's a minority of us as medical oncologists who participate, and that creates special needs. So you've got a group of individuals who spend 3 days a week in the OR. You can't be reading journals when you're in the OR. So they have an additional challenge. And I think as we go forward and we have more PARPs, we have more combinations, we have more unusual profiles of toxicities, it's going to be a challenge for the surgeons to keep up with it. I know a number of surgeons who've decided, a number of gynecologic oncologists who've decided that they wanted to do developmental therapeutics. They wanted to do chemo. And they have said they're not going to do any more surgery, and they dedicate themselves to clinical trials and these agents. Rob Coleman's an example. Rob doesn't operate much anymore. I think that's the right decision because it's a complex business. Well, I touched on it before. I think that we're going to hear about Solo 1 will probably be presented next ASCO, and then Prima, I don't know if Prima will be finished. These are both of the PARP maintenance for frontline therapy. They'll be big, and they will likely be positive, and they will move PARPs into the upfront maintenance therapy. I'm biased, but I do think trials like Forward 1, which is a randomized phase 3 trial of mirvetuximab, will be available in about a year. That will probably be a new agent. We'll hear lots of information about those combos I described, which, I don't know we really can predict whether they're going to be active or not. And then if they are active, what's the patient population? So again, the field has changed radically. 10 years ago, ovarian cancer is ovarian cancer is ovarian cancer. Now we know that it's really a series of diseases, and we're beginning to subset them. And that raises its own challenges. I mean, who do you sequence? How do you determine HRD? Do we take those patients in a different direction? So stay tuned. It's going to be an exciting time. Well, if you look at the big picture, and we can break it down, but for ovary, we do need more agents effective for platinum-resistant disease. We talked a little bit about that. Frankly, something we haven't touched on, but can someone come up with a decent early detection assay for this tumor? We diagnose all these patients with advanced stage, and so they're starting behind the 8 ball before they even get treated. So, and there's a lot of work going on that. But again, the bar on early detection assay is very, very high, right? It's got to be very accurate. But that's an unmet need. And our patients, despite the fact that we haven't really changed overall survival, our patients are living a lot longer. So the quality of life and the survivorship of our patients is becoming increasingly important. I mean, I see patients who are out 10 years. What's their neuropathy doing? Do they have sort of chronic fatigue, sexual health? All of these issues are new, and I consider it to be unmet because we haven't experienced that before. Cervical cancer, we talked a little bit about. It's less of an issue in the United States, but a huge worldwide problem. And for locally advanced or recurrent cervical cancer, it's it's still a terrible disease. We just had the first trial showing a survival advantage. First one, published in New England Journal now a couple of years ago, GOG 240, showing BEV made a difference, 3 months. So this is a disease that desperately needs something. And again, we've got these IOs, immuno-oncology drugs, exploding. It's a perfect disease for that. And then endometrial is it's a sleeper disease. What do I mean by that? Most endometrial cancer is diagnosed with postmenopausal bleeding. They get surgery and they're cured. Estrogen-driven, not a problem. But at least 8,000 cases, that's not the case, right? It comes back and they will die of the disease. And the numbers of the incidence of endometrial cancer are shocking. The incidence is rapidly increasing. Part of that is because of the obesity crisis we have in this country, which again is driving estrogen, driving increasing amounts of endometrial cancer. 15 years ago, you couldn't get a company interested in exploring drugs in this set of patients. Not only was it a relatively rare disease, you have hypertension, diabetes. This is not a great patient population. Now, that's completely changed. We have multiple companies looking at interesting drugs in endometrial cancer. And that's a good thing.